scientists at South Dakota State University are collaborating across disciplines to understand the rules of life in microbial communities at the human and animal interface.
The primary goal of my research program is to understand the rules of life as determined by the bacteria that colonize our body, specifically the gut. We are approaching this problem of microbial community assembly using a new method called culturomics. This process allows us to mimic the development of the gut colonization in the lab and do complex studies on them, through which we can understand how bacteria cooperate and or compete among themselves and with the host. The approach of using the combination of culturomics and mini bioreactor arrays to understand bacterial community assembly is really unique. This new approach are helping us to identify and isolate bacteria that never been cultured previously, and that advances the basic understanding about life. It's a platform that can be applied in many contexts that in which you can assemble simple to complex communities in a high throughput fashion that can be applied to human health, animal health, environmental studies, soil microbiome studies and such. We are starting a new project with the Sisseton Webton Oyote of Lake Traverse Reservation. The goal of this new project is to understand how changes from the traditional lifestyle of Native American people might have contributed to changes in their gut microbiome, and which might in turn would have caused chronic diseases like diabetes and obesity.
The big question that I try to address in my lab is, how do microbial communities assemble? So if we look, for example, at either humans or swine or cattle, we're dealing with essentially the same host species, so the same genetics. And if we put these hosts that have the same genetics on the same diet, we observe that there are a lot of different permutations or different variations in how they assemble. Anaerobic communities in the gut have a direct impact on the health of their host. And in animals like ruminants and other herbivores, for example, they're responsible for digesting the food that the animals ingest. So by getting a better understanding of how they work, on how they function, then ultimately we can modulate how they work. We can improve their efficiency. From my perspective, in order to address the big question, which is how do these microorganisms assemble together, I feel the best strategy is to do a comparative approach. So instead of just focusing on one host, to look at multiple hosts or multiple animal species and see how their gut microbiome assemble. When we look at SDSU, it's a campus that is very unique in the US because it has so many different animal species. SDSU is very, very convenient. Energy is a problem pretty much everywhere in the world. There's a lot of emphasis on using corn to produce ethanol. Ideally, we would want to use cellulosic biomass. And if you look at the gut of animals like cattle and bison, they were made by evolution to digest these types of products. So they represent really untapped potential to provide us with energy, but also other chemicals that we may need.
So there's a lot of interest out there right now in understanding how microorganisms colonize the human body, colonize the guts of animals and humans. And there's a relationship there. There's a relationship between those microbes and the immune systems that were developed within the body to protect the body against microbial invasion. And my work is really aimed at trying to understand how the immune system recognizes those microbes and both competes and cooperates with those microbes to benefit the overall health of humans and animals. What is really attractive about the work we're doing is that by understanding the rules written in the DNA, the genotype, and relating it to the phenotype, we're basically deciphering the underpinning relationships that have evolved over the millennia between multicellular organisms and the microbes in this world. Some of the key Broader impacts of our research include the exposure of undergraduates and graduate students to cutting-edge gene editing technologies such as the CRISPR-Cas9 technology. This is a technology that's driving forward numerous biotech applications, and so we're better preparing workforce to be able to go out and work in the biotech community and apply these new technologies. So Ben and Joy are working on understanding how these microbial communities are shaped by each other and how the microbes relate and compete and cooperate with each other. All of that's happening in the context of a host, such as the human host. The human host has evolved to recognize those communities. So there's information that's passed from the microbial community to the human host and information that's passed from the human host back to the microbial community. And my work is we're really focused on understanding the host itself and the rules written in the human host. We think that those rules are going to be different depending on the microbial community that the host is carrying. And so this is an exciting frontier to be able to relate the rules written in the DNA of the host to the rules written in the DNA of the microbes and, moreover, the microbial community.
To collaborate with these and other SDSU researchers, contact the Division of Research and Economic Development.